Hello, and welcome to the A2.3 review video on viruses. This is HL only. It's a short section, only 6 standards, and so let's talk about them. Standard 1 is all about virus structure, so the structure of viruses. They are small. They have a fixed size, which means they do not grow. Okay, so they're not considered to be living, and so they no growth. They have some sort of genetic material, and they have a wide variety of genetic material, but their genetic material has to be DNA or RNA, but it could be single-stranded or double-stranded DNA or RNA. Let's see. They have a capsid. So instead of a cell membrane, they have a capsid. That's the outer coating, and that outer coating is made out of protein. So that is the protein covering. They have no cytoplasm and few to no enzymes, depending on the type. So they're very, very simple. Not a lot going on with them. So that is what they all have in common. 2 is about the diversity. So there are differences, right? So there's different shapes. So you have, for example, bacteriophage lambda is one of the ones you're supposed to know. This is a bacteria that affects virus, a virus that affects bacteria. And it is the one that I always like to think it looks like a spider, right? So, something like this. So then the DNA is in here. And then you have the coronavirus is another one you're supposed to know. And that one's like one that looks like this. Okay. Some are enveloped versus not enveloped, or enveloped. And so that is when they have a coating on the outside or not. And so, for example, the coronavirus is enveloped. And so that means it has that layer around the outside in host cell membrane. So the host cell membrane, that's one of the things that helps it avoid detection, right? It still does have the proteins that stick out though. Those are called the spike proteins, and that's how our immune system can recognize it. Coronavirus has RNA, whereas bacteriophage lambda is DNA, and it's double-stranded. So DS stands for double-stranded. And then HIV is another one. Very similar looking to coronavirus. Oops, sorry, HIV. Looks like this, but this one is also RNA. This one undergoes the lytic life cycle. And we'll talk more about that, what that means in a second. So right now, it's 3, lytic cycle. So standard 3 is all about the lytic cycle. And so this is the active cycle of the virus. And this is when it's inside host cell, making more copies. And then it kills the host cell typically and ruptures it, thus releasing the new virus. It releases the new virus. And then that can go infect a new one, and then it will do it again. Standard 4 is about the lysogenic cycle. The lysogenic cycle is different. This is like a dormant phase, or like a hiding phase. And so this is like a dormant phase. And what it does, and this happens with the bacteriophage as well as with HIV, is what it does is it will actually integrate into host cell DNA and replicate that way. So as that cell undergoes mitosis, it's making more and more copies of cells that have the viral DNA in it, but it's dormant, so it cannot be targeted by your immune system if it's HIV, and the bacteria doesn't know, and it spreads that way too. So that is the lysogenic cycle. So this one is the active, and this is the more dormant, hiding one. Origin of viruses. There are a couple of hypotheses for where viruses came from. There are basically 3 hypotheses. The first one is the virus first hypothesis that says it evolved like at the pre-cellular stage, like before there was living things. And there's not a lot of evidence for that one. This one has probably the least amount of evidence. The second one are that it evolved from cells, and then there's the progressive or regressive. And so the progressive cell hypothesis says that it is a cell component evolved into a virus, whereas the regressive one says a cell lost things and became a virus. And so in this one, you had like a part of a cell escape and become a virus. In this one, the whole cell shrank into a virus. So instead of this leaving, and you still have the cell behind, in the progressive one, in the regressive one, that cell became one. And so there are evidence for both of these, and so there are a ton of different viruses. And so most likely both hypotheses are true. And so they're effectively obligate parasites, means they have to be parasitic. And so there's a lot of evidence that they evolved multiple times and through different methods. So this would be an example of convergent evolution. All right, so the fact that they share features is due to convergent evolution, even though it appears viruses evolved multiple times over the billion years. The last one is rapid evolution of viruses. And so they have a very short generation time. Right? They reproduce very, very quickly, which is going to lead to a rapid evolution. They produce lots of offspring. Well, lots of viruses. Viral viruses. They produce lots of viruses. So the more you do, the more you can have happen. They have high rates of mutation. And so, for example, HIV has one of the highest known rates of mutation, which makes it really hard for us to treat. And then influenza undergoes very strong selective pressure, which also is going to cause it to change because of those strong selective pressures. It also has a high mutation rate. And so you get all of these different strains, right? So we get lots of strains of influenza, which is why we need a new vaccine every year. And that is A23 viruses.